Good morning, students. Today we will discuss the difference between eukaryotic and prokaryotic cell. Eukaryotic cell, as its name signifies, eu means true, karyot means nucleus, and prokaryotic, if we split the word into two, pro means primitive, karyot means nucleus. So in case of eukaryotic cell, the nucleus is true. True means in eukaryotic cell, the nucleus is well bounded, well covered by the nuclear membrane, whereas in prokaryotic cell, the nucleus is not covered by the nuclear membrane. This nucleus is a kind of a single chromosome which is found naked in the cytoplasm. No covering is present in prokaryotic cell. Now see, first point I have written, generally the eukaryotic cells are larger in size and prokaryotic are smaller in size. Second point, true nucleus in case of eukaryotic cell. As I told you, eu means true, karyot means nucleus. Prokaryotic cell is primitive, nucleus is present in such cells. Then eukaryotic cell, they have many chromosomes, but in prokaryotic cell there is a single thread of long chromosome. In eukaryotic cell, membrane bound means covered, membrane bound cell organelles are present. Cell organelles are the cellular structures which are present in cytoplasm. So here membrane bound cell organelles are present, but in prokaryotic cell they are absent. Nucleus is also without membrane and its cellular structure, cell organelles are also without any membrane around it. Now in eukaryotic cell, bigger ribosomes are present on ER, but in prokaryotic cell, smaller ribosomes are present and they are scattered in the cytoplasm. In case of eukaryotic cell, you can see that there is nucleolus. Nucleolus is present. Nucleolus is little nucleus which is present inside the nucleus. And in prokaryotic cell, obviously it is absent because nucleus is not well defined. The genetic material of prokaryotic cell is called nucleoid. So we say nucleoid is present in prokaryotic cell and nucleolus is present in eukaryotic cell. Example of eukaryotic cell is plant cell, animal cell, that is eukaryotic cell, and prokaryotic cell is bacteria and blue-green algae cell. So this is the diagram of prokaryotic cell, that is bacterial cell. You can see here nucleoid, the genetic material is present. It is naked, it does not have any nuclear membrane around it. It is just found naked without any membrane around it and found scattered in the cytoplasm. You can see it is lying in the cytoplasm naked without any nuclear membrane. Now if you see the eukaryotic cell, the example is plant cell and animal cell. You can see here in eukaryotic cell, the nucleus is well bounded or well covered by the nuclear membrane, and inside the nucleus there is a small nucleolus which is present. Here nucleolus is not there in the prokaryotic cell, which we have seen previously. So here in eukaryotic cell, inside nucleus, a little nucleus that is called nucleolus is present. Same in case of plant cell, because both of them are the example of eukaryotic cells. Some of the gadgets or the instruments to study the cell, because we know cells are very small and they cannot be seen with our naked eyes, unaided eyes. We need some instrument to study them. They are studied with the help of microscope. It is high magnification instrument used for observing the fine details of very small objects. The types are light microscope and electron microscope. Light microscope is compound microscope and generally it is used in laboratories in schools, so it is called student's microscope also. Now a typical cell has majorly three parts. One is plasma membrane, second nucleus, and third one is cytoplasm. You see, I have given this slide. Cell organelles, cellular structure which are present in the cytoplasm are called cell organelles. Cell organelles which are present in cytoplasm, they can be divided into different groups on the basis of their outer covering. Some are having double membranes around them as a covering, some are having single membrane, and some are membraneless. If you see mitochondria, nucleus and plastids, they are double membrane, and single membranes are ER, endoplasmic reticulum, Golgi apparatus, lysosome and vacuoles. These are having single membrane as a covering around them, and some cellular structures are membraneless. They don't have membranes at all, like ribosomes and nucleolus. So starting with the plasma membrane is also called cell membrane, or it is also known as plasma lemma. Plasma membrane is the outermost delicate elastic covering of the cell, especially the animal cell, because if you see in plant cell, outermost will be the cell wall. It separates its content which is present in the inside the cell from the external environment. It is made up of bilipid layer and protein. So it has lipids, that is fat, which is two layered, and some proteins are also embedded in them, and some carbohydrates are also there. So what is the composition of plasma membrane? It has lipids plus proteins plus carbohydrate. Now after studying the characteristic of plasma membrane, next topic will be what is the role of plasma membrane. As I told you that it is selectively permeable. It allows the entry and exit of selected membrane across the cell, selected material across the cell. It gives shape to the cell. It helps in protection of the cell because it is the outermost covering. So it is also known as the mechanical barrier of the cell. It provides the mechanical barrier or the protection to the cell. Then it is flexible because it is very delicate. It is flexible, and because of this flexibility, it helps in endocytosis. It takes up the large molecules of the food. If you have to understand this point, endocytosis, just imagine about the amoeba. How does it takes the food? It extends its pseudopodia, it captures the food, and how it is possible? Because of the flexibility of the plasma membrane. So endocytosis is the capturing of the food inside the cell because of the flexibility of plasma membrane. Not only this, it allows the flow of information between one cell to another. Plasma membrane is considered to be living because later on we have to give the comparison between plasma membrane and cell wall. So cell wall is dead, but plasma membrane is living. Cell wall is permeable and plasma membrane is selectively permeable. So all these differences later on will be very helpful. Not only this, cell wall is considered to be very rigid, rough and tough, and plasma membrane is very flexible. Because of the flexibility of plasma membrane, endocytosis is possible, that is capturing of the large food molecules by the cell with the help of flexible plasma membrane. Then there are some modifications. Sometimes at places plasma membrane shows some modifications and it helps in absorption. If you remember, in digestive system you have learned that absorption of food happens in the small intestine, and inner wall of small intestine have many villi. So inner folds are there. So that is because of the flexibility of the plasma membrane, and it helps in absorption of the food. Thank you very much.